Okay, I think we're good to get started. Great. Before we begin, I'd like to acknowledge the island of Newfoundland as the ancestral homelands of the Mi'kmaq and the Beothuk. We also recognize the Inuit of Nunatsiavut and Nunatuhavut, as well as the Innu of Nitassinan and ancestors as the original people of Labrador. We offer this acknowledgement to recognize Indigenous peoples' continued relationship with their land. As biology graduate students, it is central to our education to learn about the people this land belongs to, as well as their values and their perseverance. We strive for true reconciliation and equitable partnerships, and to never cease having immense gratitude for having the privilege to work and study and call this place our home. And today, I am very excited to introduce our speaker coming in from Toronto. And I'd also like to thank Dr. Sean LaRue and Dr. Wiersma for hosting. Dr. Patrick James is an associate professor at the Institute of Forestry and Conservation. Previously, he was an associate professor at Université de Montréal and a Killam postdoctoral research fellow at University of Alberta. He holds a PhD in forest ecology, as well as a Bachelor of Science in Zoology from University of Toronto. Dr. James is originally from Paris, Ontario, where he developed a deep curiosity about forest landscapes and how they change through time. His current research in the James Lab focuses on landscape and stand-scale forest disturbance dynamics using a range of statistical simulation and empirical approaches. And with that, I will throw it over to you. Also, just a note that this session will be recorded.
Hello, everyone. Thank you very much for that great introduction. Real pleasure to be here in this very, very nice room. Thank you very much to Yolanda and to Sean for the invitation and for hosting me quite well since I have arrived. So I really appreciate it. And I'm happy to share with you our work. So I am at the University of Toronto in the somewhat strange Institute of Forest Conservation, formerly the Faculty of Forestry, established in 1907. Our forestry unit has recently moved into the landscape or the architecture and landscape design faculty. So forestry is still alive and well in Toronto, and we are growing. Today, I want to talk to you about some of the work we've been doing on insect outbreaks and population genetics. I'm kind of light on the genetics side. We're going to talk about these ideas and hoping to stimulate some questions and conversation at the end. So this is a figure that I think many of you would be familiar with and know that this is a canonical example of predator-prey dynamics and cycling populations. So the lynx hare cycle. My children really like this photo. They find it very shocking. They all get excited when I put this up. But this isn't the only system that cycles. Okay, so cycling is common across many taxa. We've got locusts in Africa, these outbreaking species. Ruffed grouse, a species of bird that exhibits population outbreaks as well, fluctuations through space and time. And what's interesting about a lot of these outbreaks is they don't just happen in one spot, but they can be spatially synchronous. So the outbreaks occur over multiple locations at the same time. My children also really like this one. This is the cute one. This is a lemming in the Arctic. And so we know this story of enormous population explosions of lemmings. And so I'm very interested in these patterns of outbreaking populations and what are the processes that lead to these outbreaks. We'll come back to this sort of at the end and try to make some inter-taxa comparisons. But for now, I want to focus on this other taxa, this other group of organisms that exhibit these really interesting spatially synchronous population outbreaks, and that is forest insect pests. This is the forest tent caterpillar, a forest pest across much of Canada, and we can see this time series, these regular outbreaks. Okay, so there's this cyclicity to these outbreaks in the forest tent caterpillar. Likewise, going out to the west coast, the mountain pine beetle, different than an erupting Lepidoptera, but it's still a species that exhibits these recurrent and quasi-cyclic outbreaks through time. So this is some historical data going back to the 17th century and showing that there is this history of recurrent outbreaks. And so the questions that we have is like, well, why do these outbreaks happen when and where they do? And what drives this periodicity? Why don't they go up? And why don't they just stay at a high density? Or why don't all taxa exhibit this sort of massive fluctuation? And when these outbreaks happen, this isn't just a slight increase in the number of individuals. We're talking about multiple orders of magnitude increase in population density, and then likewise a crash that's followed. Perhaps more germane to our Eastern Canadian context is the spruce budworm. So this is a native Lepidopteran forest insect pest that exhibits these regular cyclic outbreaks. This is a really great study that was, or the image here is from a great study by Boulanger and Arsenault that goes back to 1500, showing that there is this record of outbreaks. Was that an echo?
Or was that?
Oh, it is an echo.
It's an echo.
No.
Was it that microphone here?
hearing me?
Sure.
Deepal, do you have, or
Am I still echoing?
Still echoing?
No.
Test. Sounds good. Okay, thank you. Nice fix. So this cool figure that shows the history of outbreaks of spruce budworm in Quebec, and they used this dendrochronological technique, meaning they took tree cores, and they were able to go back this far in time. They went and they found churches in areas that were in the range, and they took cores of the churches, knowing that the churches would have been built from wood nearby. And so those trees themselves would have been 400 years old when they were cut down. The church is 400 years old. So you have this, you can reach into the past with this dendrochronological study. So regular, recurrent cyclic outbreaks, and then more recently, the last 3 outbreaks with great regularity, and each outbreak larger than the last. We care about these outbreaks because they compete with humans for access to resources. Right? So these bugs eat the trees we want to turn into pulp, paper, and 2x4s. These periodic fluctuations, our interest in them is not new though, right? Charles Elton wrote this incredible paper in 1924, so coming up to 100 years. If you're interested in population dynamics, animal or otherwise, I highly recommend looking at this paper. And he specifically was focusing on the lynx hair cycle and was relating it to the sunspot cycle. And for those of you who might have looked into lynx hair stuff, would know that sunspots were fashionable for a while, and then they were incredibly unfashionable, and now they are somewhat fashionable again. And so these models or these theories that helping us understand outbreaks change through time. So Charles Elton was writing about population outbreaks and cycles and what drives these spatially synchronous cycles almost 100 years ago. And he did take a moment to talk a little bit about insects, right? Talking about this increase of individuals and migration and destruction of resources here in the forest. And because, as I said in the title, I'm interested in talking about population genetics, Charles Elton alluded to some of these genetic consequences of outbreaks in referring to the constitution of populations that survive the outbreak being relatively or comparatively pure. So I think he's talking about, we can map that onto bottlenecks or successive bottlenecks and reduction in genetic diversity. So what does that look like through time? So back onto the spruce budworm. This is the framing, and this is sort of the context that makes me very interested in these outbreaks of spruce budworm. This is the expansion of the outbreak in Quebec, and it has touched other areas in Eastern Canada. I was working in Quebec at the time, so this is the data we have. And I showed this animation up until I think 2018, which is when the postdoc left the project, so we didn't update the map after that. But we see this spatially synchronous development and growth of this outbreak. And so the question we are interested in, or that I think emerges clearly from this image, is like, well, why do they happen all at the same time? What is connecting the spatial processes at these different locations? Is it that all of a sudden all populations decided to rise simultaneously like a tide floating boats? Or was it one location started to exceed some sort of population density and sent migrants to other locations, sort of seeding those outbreaks in other places? Was it a combination between the two? And distinguishing between these two ideas is actually quite important for how we approach management of these resources. If it's one versus the other, can determine what type of management approach we might use. So I can outline these two contrasting theories, whether it's boats floating or dispersal driving the outbreak. So if you look at this image and imagine just a flat space across the bottom, populations A and B, the height of the green bump is the population density, and the red is detection threshold, when trees start to die or change color, and we can see what's happening. So in the one theory, population B exceeds the detection threshold, and we would call this an epicenter, much like we see on these defoliation maps, which exist across Eastern Canada. So these red points would be an epicenter. And then it produces migrants, which then disperse to other locations and encourage that other location to exceed the detection threshold, right? This is dispersal. This is flatten the curve. This is wear a mask to prevent dispersal of COVID, right? The other option is that there's some sort of spatially autocorrelated weather, environmental conditions that cause all populations to rise at the same time. And this is referred to as the Moran effect or the oscillatory hypothesis. And as I mentioned a moment ago, distinguishing between these two strategies is really important within an early intervention context. And I think some of you might know about early intervention. If you don't, this is a current framework that's being developed, has been led by researchers at the CFS in New Brunswick to better control outbreaks. And it focuses on the idea of identifying incipient hotspots of the outbreak and spraying them, in this case with BTK. This is an image of a plane spraying DDT in the 50s. And fortunately, we don't use DDT any longer to control these outbreaks. But we're still using this notion of, can we identify where the populations are developing, and then can we control them with precision application of pesticides? If everything is rising simultaneously, early intervention is unlikely to help us. If an individual location is producing dispersers that then infect other areas, early precision application of pesticides can help control the outbreak. So to ask this question, to distinguish between an epicenter versus an oscillatory hypothesis of outbreak dynamics, we opted to take a population genetic approach to it. So the lens through which we want to see this was on spatial variation and genetic information, specifically using gene flow to infer dispersal. So if we can identify significant patterns of dispersal, we can start to find weight of evidence in favor of one of these two hypotheses. I'm not going to go into a whole lot of detail here, but suffice it to say that we can estimate gene flow or the degree of connectivity between populations using similarities and differences in allele frequencies between locations. If allele frequencies are similar between locations, we assume there's some degree of connectivity. If they're quite different, we assume that they are not connected, right? So we expect spatial signatures to differ depending on the relative importance of dispersal in the outbreak system. So just quite visually, we have two populations with very little dispersal between them. The colors indicate that they're different, and we can identify this on the basis of allele frequencies. If they are connected, they start to be mixed up, and we can't distinguish them, and that would say, well, there is movement between these, and that would be evidence in favor of more of a role of dispersal. And parenthetically, these butterflies are actually beta symbols that I use. It's just text, and I rotated them and stuck them together. So if you ever need a quick shortcut to make a butterfly for a presentation, you can use a beta symbol. That's yours. Please take that. And so this was the context. I got roped into this first project to understand spatial population genetics of the spruce budworm system. I was talking about mountain pine beetle last week. I'm talking about spruce budworm right now. So talking about the spruce budworm, I was working with a guy named Brian Sturdivant, who's a researcher at the US Forest Service in Rhinelander, Wisconsin. And he developed this really great project based on this really cool study region that borders Ontario and Minnesota. And we call this the Border Lakes landscape. And it's interesting because this project was actually focused on forest management legacies in particular, because forest management in Ontario is different from that in Minnesota. And likewise, there's Quetico Provincial Park, which is an enormous wilderness area in between the two that has no history or less history of forest management. So as he designed this project, and I got to go and help out with fieldwork, we identified a developing outbreak here in northern Minnesota, and that's indicated in red. And we drove around and we sampled spruce budworm at all of these locations. And we wanted to know, so even though there was no outbreak here, we could find individuals at low density. And we wanted to assess how similar were the individuals where this developing outbreak was to these other locations. We did, my role in this project, we actually canoed through Quetico and paddled into the United States. So we arrived by canoe and we collected budworm all the way through on this trip, about every 15 kilometers. We arrived at US Customs and they said, okay, what's the number of your vessel? And it was a canoe that we'd rented in Canada and the number was 5. So it was like boat 5. And they didn't like that. They were quite unhappy with us with that regard. But we paddled through, we collected moths. We didn't use them for this study in the end, but it was a real 2-week adventure paddling through Quetico. So in the end, we had about 230 individuals. We're using microsatellite loci, these 7 sites. And we considered the larvae that we collected from individual locations to be our residents, like a local indicator of genetic variability, like with some fidelity to that location. And then we also collected moths from pheromone traps at the same location. So the moths could have been local or they could have been migrants. And so we sought to use our genetic information to distinguish these residents or ask the question, can we distinguish the moths from the residents? Are they similar? Right, as I'm asking here. And so the first thing we did, and this is maybe a bit of an abstract graph, but we did some ordination, right? So multivariate genetic data can be treated as community data. We can use community ecology methods. So this is a principal coordinates analysis. And if you don't have experience with this, all this is if you can imagine a matrix where each row is an individual and each column is a locus. And so it's this big matrix and we can collapse that multi-dimensional data into just 2 dimensions and we can visualize differences. So points that are closer to one another are more similar with regards to their genotypes and points that are farther away are more distinct. And what was exciting about this is that we were able to find, pardon me, that the larvae exhibited some sort of fidelity to the sites, right? So we had good spread with the larvae. They all sort of represent their own spot within that ordination space, whereas the moths all clustered together. So the moths all came from one location and the larvae were all from their own location. So what we're seeing here, just with this simple within season, 1 generation plot, is a significant amount of mixing in the population. So the moths are all coming from just one location and the larvae are their own. We produced some other images, some other plots. This is a structure plot where each bar represents an individual. And the color represents the probability of assignment to different groups. You know, using this Bayesian model, we can identify the number of groups within our sampled data and then assign each individual a probability to that group. And so there were 5 groups and the color indicates that probability. And so we can see within this area, we had these 5 groups. And then we can also organize them by site. And you'll see that each site was kind of mixed up, right? So no sites were really clearly one cluster or the other. And this was a bit confusing at first, but we took a really simple approach and we just did a chi-squared statistic to see whether these groups and sites were independent or related to one another. And sure enough, we were able to find some sort of support such that, you know, group 5 was associated with site H. So that was its own thing. And group 2 was associated with site G. And then site I was this mixture. And so what we were sort of finding was that we had this legacy of the previous outbreak that was found everywhere. And then it was being mixed up with these unique genetic signatures or that unique genetic signature from that outbreak that was occurring. Okay, so we had some sort of structure that was latent from the larvae that was being mixed with the moths that were dispersing. So within 1 generation, we're seeing this incredible amount of mixture and dynamics. You know, and we're using the genetics here as a proxy for population connectivity and movement, right? So this is evidence in favor of a great deal of movement and complexity in the system. And so what we take away from this, or what I'm arguing in favor of, is that ecological context influences genetic inference, right? And with an outbreaking population, the type of answer you get is going to depend on when you measure it, when you sample it. And by extension, perhaps other systems that exhibit sort of population fluctuations or outbreak dynamics. If you want to make inference about what's going on in that population, it's going to depend on when you sample it. So in this figure, we show just a hypothetical outbreak, right? We go from a light outbreak, moderate to a severe. And then these different panels look at what genetic inference might look like depending on when you sampled it. So if you sample it at the outbreak peak, you're going to find no differentiation, and you might conclude enormous dispersal capacity, high connectivity in this system. Whereas if you sample it during an intermediate stage of the outbreak, you might find, oh, intermediate levels of connectivity, intermediate levels of movement. Versus in the low endemic phase, when populations become very low and there's very little dispersal, one might infer limited dispersal, limited connectivity in the system. So these dispersal connectivity attributes are dependent on ecological context. And likewise, you could find multiple within the same landscape. So ecological context influences genetic inference, and it's something we need to be aware of, I argue, when examining populations that have temporal spatial variations in density. So what? This is the rude question I always ask my students, and so I ask myself, so what? Why do we care? What I think is exciting about this is with these outbreaking systems, highlighting that demographic context can influence inference. And it shows, I mean, in the context of this ecological theater and evolutionary play from Hutchison, you may have seen that in the past. The notion is that this theater is actually dynamic itself. And in a way, I'm just trying to flip the script a little bit and put the ecologist in the driver's seat for a little bit alongside the evolutionary biologists. But this ecological theater itself is dynamic because of these cyclic population dynamics, these rapid demographic changes. And importantly, functional connectivity is dynamic over the course of an outbreak. So I did this work just at the end of my postdoc, and I moved to Montreal. And in Quebec at that same time, there was this great outbreak developing. Great as in, for me, not so great for the balsam fir and white spruce in this landscape, but this really exciting opportunity to look at this spatially synchronous outbreak that was emerging. So what year was this? This was 2015. So I arrived in 2012, so the outbreak was well on its way at this point. So I wanted to test those two hypotheses I described before in this system. So is it epicenter or is it oscillatory? Can we use population genetics to find support for or against one of these hypotheses? So we focused on these different patches. If it were the epicenter hypothesis, we'd expect all these patches to look similar, right? Because they would have all come from one initial outbreak location. Conversely, if the oscillatory hypothesis were driving the system, we would expect them to look different genetically. So we went to the field. I hadn't done much field work before, and then I was in charge of running field work. So this was an interesting experience, and we had the opportunity to visit some incredibly beautiful landscapes in northern Quebec, learn sort of the upper limits of the amount of windshield time a graduate student can sustain. 1,000 kilometers a day is too much, so we don't do that anymore. So some big distances to travel, lots of bugs, of course, and we collected larvae. And it's a nice system to work in because when an outbreak is happening, you can just walk up to a tree and find these caterpillars just hanging out on the tree. This is a somewhat outdated map of all of our sample locations for which we have spruce budworm larvae across eastern Canada, and it does not touch the information we have in Newfoundland and Labrador, Nova Scotia, New Brunswick. The stuff down here in Maine, oh, pardon me, New Brunswick is included. Stuff in Maine was contributed by collaborators. They mailed them to us. And so from 2012 to 2015, we collected many spruce budworm with the objective of sequencing them and testing those hypotheses. So in total, a little over or around 1,000 individuals. And after a whole bunch of filtering, we had about 600 SNP loci. This was using a genotyping by sequencing approach, no reference genome at this point, so just going through platypus and stacks to align those fragments. I'm happy to talk more about the bioinfo details if anyone has questions at the end, but that's as far as I'm gonna go here. So we had 1,000 individuals, a bunch of SNP loci. And we sought to understand variation in genetic diversity through space and through time. And so this is a work of a clever postdoc who worked with me for a little while, Jeremy Larocque, who's now back in Germany. And we started with mapping simply heterozygosity throughout this landscape. So this is 2012. And we can see that some sites had high levels of diversity and some sites had low levels of diversity. And so this was interesting. This is where the outbreak had started. And so there's high diversity in this outbreak core. And then in these peripheral areas where the outbreak really hadn't quite got going, there were lower levels of diversity. And then through time, 2012, 13, 14, 15, and unfortunately just logistics, we didn't get the exact same network. We see that the diversity homogenized. So those areas of low diversity became high diversity and became more similar to what we found in the high density locations. So this was an interesting first result without anything high powered or anything complex in terms of analysis, just spatial patterns and diversity that we had an overall increase in heterozygosity and a homogenization across the landscape, which we initially took as strong support for the role of dispersal in this outbreak, because they're not going to get this increase in diversity without the influx of other individuals. And around that time that we were trying to make these inferences about population movement and connectivity using genetic data and all this bench stuff and statistics, this was coming out in the news, right? So the outbreak was really getting going. And there are these massive, this is from the inside of someone's vehicle looking out, and this is their windshield covered in moths, right? And so moth infestation, likely spruce butterworm. They didn't check a taxonomist. It was definitely spruce butterworm, right? Like no other species is doing this. And you can just find these heaps of moths on the side of the road. So these population outbreaks are just incredible. There's a photo that I sometimes include, and I forgot to include, but I'll describe it for you. In northern Quebec, there are many dams, right? Like very large dams, and they're all lit. And there's a photo of a front-end loader that seems to be picking up sand, like a full-size front-end loader picking up sand, but it is a heap of moths at the base of a light at the Manic-Sank Dam north of Baie-Comeau. So huge outbreaks. So like I said, we're trying to make inference and being like elegant, delicate with our genetic data, and so we can just watch this unfolding. And even further, you could see it from radar, like just the nature of these outbreaks, the scale, and this is why the system gets me so excited, because these tiny organisms that when they're not outbreaking, you can't find them. They are impossible to find. And here we are on, what is the date? July 15th, 2013. Blue is snow, supposed to be. Oops. So that is a massive cloud of spruce budworm leaving the North Shore into the South Shore. And then the next day, the pattern went right back. So these huge clouds of moths, all moths, in the millions. And so, I mean, our colleagues at the CFS who are using this radar data, we're like, oh, look, we found some genetic evidence for high connectivity. It's like, bye, voila. Like you can see it with your own eyes. Anyway, so it's important to have complementary sources of information when telling an ecological story, and this is one of them. We also did some other analysis on these genetic data, like classic isolation by distance plots, right? And the story here is that there was some structure in 2012, and beyond that, we lost all signals of structure. So it seems like at 2012, when we were first examining this population, there's still some remnant, some legacy of the previous outbreak collapse, and this is captured by these sort of two groups here. So there's some differentiation, but mostly it was all a wash. And just to remind you, this is on the order of 1,200 kilometers. This is 1,000 individuals. No differentiation at these massive scales. So a bit of a frustrating outcome for a lot of time and energy into sequencing data. One hopes to find some sort of structure, some differentiation that we can talk about, but it conforms with our hypothesis, our theory that as the outbreaks increase and populations increase and dispersal goes crazy, everything becomes really connected. And so these data bore that out for us quite well. We then wanted to look a little bit into New Brunswick. We were working with colleagues at the CFS. They were curious about the individuals that they were able to identify in New Brunswick, were they coming from Quebec, or were they their own population that had not yet increased? And so we took a subset of our data in Quebec, and we sampled a whole bunch of individuals from New Brunswick. And this was an interesting challenge as well, because like I said, in Quebec, we could walk in the woods and pull these big, juicy larvae off the trees with great ease. Whereas in New Brunswick, where the outbreak had not happened, it was a much more elaborate process that the CFS took care of sampling branches and washing them and rinsing them. And this is a Petri dish, and this is a zoom in of the Petri dish, and that is a second instar larvae. So incredibly time consuming, incredibly difficult to find. So every one of these blue squares, diamonds, is the location of an individual L2 that was found for us, which we then also sequenced, which was its own adventure. So very small, very detailed. And we were asking the question, are these guys similar to these ones, or are they separate? Is this an outbreak? It's not an outbreak yet. Are these populations disconnected from what is actually just a complete tidal wave of budworm on the North Shore? And we were able to identify that there was some incursion. So this is what is this? This is I forget the name of this analysis. It's a Bayesian clustering method which uses spatial priors. So it's a kind of a nuanced method to identify the structure. It's a bit more detailed. These effects are a bit exaggerated by the color bands that are used. The differences are quite subtle, but they were indeed present and significant. So there was one location that seemed to be related to the population in Quebec, but for the most part, population in New Brunswick was independent of the massive outbreak in Quebec, which gave them and continues to give greater justification for this early intervention approach. And anytime a hotspot does emerge in New Brunswick, would be to apply the pesticides, suppress that population. And New Brunswick is having and continues to have great success in controlling the outbreak. They have not experienced the outbreak that the Gaspé and the North Shore of Quebec has experienced. We believe attributable to this quick action with their early intervention. So complex spatial patterns, interesting dynamics, and seems like dispersal does play a really important role in these outbreak dynamics. I'm not sure how I am for time right now. Great, thank you. I'll shift gears a little bit and talk a little bit about the context of these outbreaks. And something that stands out are these cyclical outbreaks, these great spatially synchronous dynamics, and the questions that come out of that is, well, why does it do that? And we talked a bit about this with some students this morning. Why do these outbreaks occur? And the general framework of population outbreaks is a tritrophic model, right? We've got 3 trophic levels. We have the host, the spruce budworm itself, and the predators, all of which is moderated by some sort of climate, weather correlated function. In working with the spruce budworm, we're very interested as well as in the population of natural enemies, the predators, which are thought to play a really important role at some parts of the outbreak. And this network of natural enemies outside of the context for the outbreaks is incredibly fascinating. It's incredibly rich. This is the full food web that is associated with the spruce budworms. We've got the spruce budworm, Cristanura fumifera right here in the middle. Here's balsam fir. And then this massive network, connected network of parasitic wasps and flies that attack the spruce budworm. Now, all these species aren't active at the same time. This trophic network changes through the outbreak. So early outbreak has one subset of species, mid-outbreak another subset, but this is all of them all at once. We wanted to understand some of the spatial variation in these natural enemies, and what does that mean for outbreak dynamics? And we focused on a couple of species of wasp, in particular, a species called Apantales fumiferana and Glypta fumiferana. Shows you some of this work. This is a spruce budworm larvae. This is a Glypta larva that has emerged from it. So very much like alien, these wasps and flies attack the larvae. They lay their egg inside the caterpillar, which consumes it from the inside out and then emerges. Only a single larva emerges from a single budworm. unlike other parasitized species where multiple caterpillars can emerge, there's actually a really fascinating like competitive dynamic that occurs within the budworm larva. You can see these early instar forms of these things look like, I don't know, like sandworms from Dune. And they fight and consume each other inside the body of the spruce budworm such that only one emerges. So, students doing some work identifying parasitoids, and this is the, just an example of the parasitoids we have. So we have a full community database. We reared about 100,000 spruce budworm larvae individually in synthetic diet. And we have now identified successfully 9,000 parasitoids through a combination of taxonomic stubbornness and some barcoding work to have this data set, which is yet to be analyzed. So if anyone is really excited about community ecology and budworm parasitoids, we have this enormous data set that needs a clever person to work on it. So Simon Legault, who's a PhD student in the lab who's just finishing up, he wanted to do some work asking about the population genetic structure of these two species of parasitoid and how it relates to the population genetic structure of its host. Right? And knowing that these species are different, we know very little about the parasitoids, whether they can disperse really far, how much they disperse relative to the budworm itself. And so we sequenced 300 some odd glypta, 279 epanthelles at 1,000 loci across this network of sample sites. So his question was, what is the spatial structure? And what does it look like relative to the spruce budworm? And I'll remind you, the spruce budworm, we found no spatial structure, 1,000 kilometers, no differentiation. It's a bit of a bummer. Once again, completely flat. And again, this is a tiny wasp, scale of thousands of kilometers, no relationship with distance. And again, this is genotyping by sequencing, no reference genome, 8 times read depth. This is good quality genomic information showing no spatial differentiation, no isolation by distance in the system. And we were a bit surprised by this. We didn't expect to find it. Likewise, using clustering approaches, no evidence of clustering. So not genetically homogeneous, but very little or no differentiation across these enormous spatial scales. So again, a lot of work, not much of a result. He worked very hard and we argued hard as to why this non-result was incredibly important to report, and we were successful in doing so. So just a highlight and a reminder that results that don't conform to your hypotheses or results that have p-values greater than 0.05 are still worth publishing, and you should fight for those results so that other people don't waste their time in the future trying to answer the same question. One trend that Simon did identify were these spatial trends in heterozygosity. So after sort of the bust of trying to identify isolation by distance or differentiation, we're asking about trends in heterozygosity on a latitudinal gradient to see if we could see sort of like range edges, whether different species were approaching their range limits as we went northwards. So we plotted out epanthelles and glypta. This is latitude. This is the expected heterozygosity. And we can see a decrease in the heterozygosity for epanthelles, the smaller of the two, and a flat relationship with glypta. So it's suggesting that epanthelles might be reaching the northern range limit, and that glypta itself might be the more northern species of the two. And this has consequences for understanding northward range expansion of these outbreaks due to climate change. We're finding, the climate's warming, we're getting more susceptible species further north. The budworm is appearing farther north than it has before. If the natural enemy community differs in these more northern areas, the outbreak outcomes may themselves be different. So this is just dipping a toe in this arena. He's continuing to work on this idea, but there may be fewer natural enemies at the northern range limit, which could have consequences for outbreaks in the future. So I'll just talk quickly about some ongoing work, and before I wrap up, continuing in this landscape genetics or this genetics world with spruce budworm. Very interested in how range expansions and outbreaks affect our ability to make inference. Postdoc in the lab, JP Fontenelle, is asking some landscape genomics questions and asking how much the outbreak influences our ability to identify potentially adaptive loci during the outbreak as well. So here we have year. Here is the number of outliers that are detected using 3 different outlier detection methods. And in a way that was really similar to the heterozygosity we saw in the spatial patterns and neutral genetic diversity, we see this peak in putative outlier loci in 2014, which then decreases, which suggests to me that a lot of these are false positives that are the consequence of the spatial dynamics of the outbreak. I mean, the punchline here is that these range expansions mess up all these methods and cannot be trusted. They should not be trusted. And because a lot of ecology is outbreak and spatial expansion, we should be quite suspect of these tools, or at least find ways to integrate these spatial dynamics when we're asking population genetic, population genomic questions. And we didn't make up this problem. Lawrence Escoffier and Ray in 2008 talked about genetic drift and allele surfing during range expansion, where simply through stochastic sampling processes, you can get clines developing in systems as the outbreak, as a population expands, clines and allele frequencies can emerge. So, one visualization, this is a simulation they ran where they had an advancing front and a mutation was initiated at this location. And you can see through only neutral spatial processes, you have this cline in allele frequency that's generated. And this is what we hypothesize is driving some of these spatial patterns during the outbreak. As they expand, we're getting these biased patterns that are confounding tools and methods. And why I talk about this, and I think it doesn't only apply to the spruce budworm world, but I think it's applicable to studies of invasive species as well. If we're trying to identify loci that might be influencing the success or this establishment of invasive species, using these methods, landscape genomics, or trying to use landscape genetics, population genetics to understand these dynamics, they might be biased simply by these spatial processes. JP is also working on some simulations. We've developed a individual-based demogenetic model. So we track individuals. Each individual has a genotype. They move around the landscape subject to a whole bunch of rules, and they recombine, and we can see the consequences of different assumptions about dispersal, connectivity, effective population size, and what that means for emergent landscape scale population genetic structure. So the objective here is to compare our simulated spatial temporal genetic patterns to what we observe, and in a way try to back engineer the ecological conditions that may be driving the spruce budworm outbreak when we can find a match between the simulation and what we've observed. And finally, our next step, and so I've got Newfoundland in this figure, our next step is to leverage other extensive existing data sources and to expand our sequencing efforts to better understand the genetic consequences of these outbreaks at a broader geographic scale. And so we're leveraging data that have been collected in the context of the Budworm Tracker Program. This is based out of New Brunswick. It's a large citizen science project that sort of overcomes some of those windshield time limitations of traveling to all these locations and collecting individuals. And what this program does is it mails a small pheromone trap, so a small pheromone-baited trap to citizen volunteers. And this is the extent of all of the locations where these traps have been sent. And these volunteers, they check the trap every other day, and with great enthusiasm even, and send back information about the time series of moth arrival. And they send us the moths, we get demographic, timing, and then genetic information as well. So these moths from 2014 to 2013, an enormous data set that we're going to be mining. These moths are all in freezers in Fredericton, and we plan to go in the coming months to sort of sift through and pick an optimal subset to capture the spatial temporal variation, and then further refine some of these population genetic studies that I shared with you already. So the key takeaways here is that I think demographic context influences population genetic structure, and we should keep that in mind. The genetic structure of the spruce budworm, at least, changes as the outbreak progresses. It's not an intrinsic feature of the population, the things we're identifying. It's something that's transient and related to demographic context. Shockingly and somewhat disappointingly, there were no barriers to parasitoid connectivity over thousands of kilometers. Genomic outlier detection methods can be confounded by outbreaks and range expansions, so we should be careful. And there are lots of exciting questions that remain to be answered. And so I'll just bring it all the way back like I threatened at the very beginning of the talk. So we've got these different systems that exhibit outbreaks, population fluctuations. Are they all governed by the same processes? So, we were talking a little bit about epigenetic shifts. Some populations show these changes to be hyperfecund, and that translates into an outbreak. Which outbreaks are ecological? Which are genetic? How do we distinguish these things? I think there are commonalities, and from an eco-evolutionary perspective, I'm increasingly curious about what common selective pressures these populations, these species have experienced. Are they all coming up with a similar solution to a similar problem? And then I'll go out on a limb and suggest that trees are also sometimes outbreaking species. So spatially synchronous masting is kind of a plant equivalent of an outbreak. And a lot of energy and time has been spent in describing the evolutionary pressures that drive spatially synchronous masting in trees. And the synthesis, as far as I understand, is usually for satiation of natural enemies and predators. Is that the same strategy is that the same pressure that has driven these really complex animal population dynamics? So I'm hoping to sort of move into a bit of this realm and ask, borrowing some of the techniques that have been used to understand masting in trees and apply them to my preferred outbreaking insect species and try to better understand the evolutionary forces that shaped these rather extreme life history strategies. So with that, I'll thank NSERC, Natural Resources Canada, FQRNT, and the Atlantic Canada Opportunities Agency for funding for all this work, as well as the graduate students and collaborators who have helped us with this. And I'm happy to take any questions. Thank you.
Just have to go through
Sure.
speak through this to answer. All right. Question.
Has there been collaboration between bird species, or bird increase or fish increase due to the outbreaks? Like, have the populations changed? Is there collaboration that's happening with that?
It's a great question, and there has been a lot of interest in how bird populations respond, and there's a suite of warblers, a specific suite. And if you're a bird person, please forgive me for not getting them exactly correct, but I think it's chestnut-sided, Tennessee, and Canada warbler are three species that are identified as budworm specialists, and they exhibit this numerical response to the spruce budworm for sure. We recently did a paper trying to harness these increases in bird population density as an early indicator of the outbreak. So before you can see the defoliation, does acoustic monitoring indicate an increase of these populations that could identify where you might want to survey or treat more with a bit of a earlier intervention? So yes, there's a bird effect. It seems to be limited to these few species of warbler. With regards to fish, I know less. It's funny, I reviewed a paper on the way here on the plane looking at carbon and the sources of carbon in different species of fish following an outbreak, and that there are these biodiversity cascades, and that the main sources of carbon seem to change as the outbreak occurs. But all of these would be, they're pulses, right? Like they're this ephemeral change. And I think understanding the nature of the duration of that ephemeral pulse is still a really interesting question to be asked.
I think there's a couple questions online. Okay, Louise is asking, how might BT spraying have affected your genetic outcomes?
It's a good question as well. So if one were to sample a location that had been sprayed, it could result in the elimination of the local genetic variation. And then we would be more biased to sampling recent immigrants or dispersers into that location. Much of the area we studied, at least in Quebec, was not sprayed. We were in sort of extreme isolated areas. But yes, I don't exactly know. We could hypothesize, and it typically doesn't kill the entire population. Like there's like some that remain. Sorry, I can't answer that any further, but it is a question worth considering.
Any more questions from the room?
Hi, Patrick. Thanks for your talk. So you talk about the two competing hypotheses about spatial synchrony. And I'm curious if, so in a severe outbreak situation, you mentioned, you find that there's low genetic distance between individuals, pointing you towards the dispersal hypothesis. And I don't know much about spruce budworm and their generation time and anything like that, but is it possible that there is sort of a Moran effect that's causing these spruce budworm to display a similar genotype without that dispersal? And how do you control for that? How do you make sure that's not what's happening?
Yeah, I think that we have to wait for the outbreak to either start, well, perhaps collapse and go down to these lower densities, right? Because right now densities are just so high and it's panmictic. So we actually don't have the information right now to distinguish that. We continue to monitor, and with this budworm tracker data that I demonstrated, it is possible that we could then add a lower density when there is less connectivity, begin to tease that apart. So even in Quebec, we've continued monitoring every other year. It's a univoltine species, right? Like insects are convenient to work with because it's just one generation per year. So I think we'd only really be able to tease that apart at lower population densities when they aren't completely mixing. And so if we were to find variation in densities and changes in population densities that are synchronous without dispersal at lower sort of regional densities, we could get a handle on that. But it's, yeah, for the future.
Thanks. Whoa. Yeah, great talk. I'm wondering if like population sampling ebbs and flows with outbreaks, where like when there's an outbreak, we're really interested and we go out and we do a whole bunch of sampling. And then when there's not an outbreak, there's less of it, because it seems like there's some really interesting questions that you need to do some extensive sampling at low densities. So I'm wondering if that, I don't know, in your experience, is it fairly consistent, or are we missing a lot of low density information?
Right. So, I mean, much like human epidemics, like monitoring is key. Unfortunately, there hasn't been as much monitoring at low densities. We know more about peak outbreaks, and if I can like digress a little bit, that's a function of the lag in agencies and funding organizations, and they only release the money when enough voters are upset enough to demand that. So there's a lag. So it's very difficult to get money at low density population times. And that's not an excuse. It's sort of just an explanation of what's going on. There are some other data types. There's at least in Quebec and New Brunswick to some degree as well, there are annual L2 surveys, which is these the L2 densities. So people go, they cut these branches and they do it regularly. And there is some monitoring of low population densities, but it's not a regular network. It's this fascinating data set that sort of tracks the potential outbreak. So it's this heterogeneous point data set. So what we're trying to do with monitoring the outbreak as it collapses is to fill the very gap you're describing. So even without funding on a shoestring budget, I can drive up into northern Quebec with a couple of students and spend a week camping and collect larvae and bring them back. And then when when the money taps are turned back on again, we can do the sequencing. To your original question, there is a gap in the endemic stage monitor.
Yeah, thanks.
Any other questions from the room before I take one online? Hey, do you see any evidence of syncing between your budworm cycles and Elton's classic cycles over the same time span?
So the sunspot sort of cycle question. We've not yet investigated it. I think it would be something that would be interesting to explore. But there are these other cyclic patterns as well, you know, like ENSO and El Nino, these other larger scale weather dynamics that could be involved in synchronizing these outbreaks. We know that they play a big role in fire weather and those outcomes. So it is very plausible that they would relate to mudworm as well. In a way, I kind of avoid those questions of what drives the cycles because it's a really hard question with a lot of people that have worked on it. I mean, Robert May worked on these sorts of problems way back when. It's a very crowded field. Yeah, I think to the person asking the question, that is an excellent question. I would encourage you to look into it.
Okay. There's no other questions. We can stick around for a little bit in the room and have some snacks and coffee, and anyone can come and have a chat. But yeah, thank you everyone for coming.